primer desafío y más importante desafío es en el año de 1997, en donde los biólogos Kirk Campbell y Jan Willmar presentaron la proyección, la mostraron al mundo la oveja Dolly, una proyección enormemente compleja en donde aparecía Dolly como una proyección científica en donde era un clon y ese clon estaba creando una situación enormemente e indescriptible, además, de poder entender y comprender la comunidad en general de qué se estaba hablando. ¿Qué estaban hablando los científicos? ¿Qué era un clon? ¿Qué era lo que sucedía con ese clon? ¿Y cuáles serían las consecuencias posteriores? Era realmente algo inadmisible e incomprensible definitivamente. Al mirar esta situación científica que se estaba presentando en el 97 y hablando con mucho detenimiento con el rector del Antonianum de Roma, el doctor José Antonio Merino, franciscano, en donde los franciscanos tenían y tienen que ver definitivamente con el desarrollo de la vida de una manera muy especial, dadas las características mismas de la proyección franciscana con San Francisco de Asís, de hablar de la naturaleza y de hablar con la vida, y hablando los dos con mucho detenimiento, encontramos una situación muy compleja de poder comprender hacia dónde caminaba el desarrollo científico. Y decidí fundar una cátedra que pudiera tener la comunidad respuestas muy complejas, pero muy importantes en la misma toma de decisiones de lo que a partir del momento de la oveja Dolly vendría en el campo de la vida, de la naturaleza, de la toma de decisiones del ser humano. Y surge la cátedra en el año 2000, cuando es presentada justamente la oveja Dolly, y conjuntamente en la presentación de la oveja Dolly hacemos y fundamos la cátedra en la Universidad de Monterrey. ¿Qué fue lo que me motivó? Indiscutiblemente el que el ser humano, la comunidad, pudiera tener respuestas prácticas, concretas y accesibles a su mundo de ese momento, en donde estábamos encontrando situaciones enormemente complejas de entendimiento. Porque veíamos dos problemas muy severos. Uno, el problema del avance de la ciencia, y otro, de la concepción y la decisión de la ética. ¿Qué le correspondía a la persona humana, al ser humano, frente a una serie de situaciones enormemente difíciles en ese momento? Y difíciles en ese momento, pensando también que ahí no se quedaría toda la parte biológica frente a la proyección de un clon como era Dolly, sino que el avance de la ciencia iría llevando de la mano al ser humano a una serie de características y de posturas en donde tendrían enormes cuestionamientos, como lo ha sido hasta el momento. Hasta el momento, a partir de ese momento de un avance tan extraordinario, pero tan complejo de comprender, sin duda alguna, para el ser humano se vio y se ha visto en enormes cuestionamientos a partir de ese momento. ¿Qué es lo que se puede hacer? ¿Qué es lo que se debe aceptar? ¿Cuál es el raciocinio de cada ser humano frente a toma de decisiones personales, eminentemente personales? Lo estamos viendo hasta el momento. Y eso hizo que la cátedra tuviera una característica enormemente fundamental y un gran reto. Invitar a un científico en el campo del desarrollo de la ciencia e invitar a un ético en el campo de la toma de decisiones de carácter personal y lograr que una institución absolutamente fundamental como es el Vaticano pudiera planteársele nuestro reto o mi reto, como quieran ustedes verlo, y decir, necesito de la ayuda de ustedes para que podamos darle respuestas al ser humano en el campo de la ciencia y en el campo de la ética. Y se partió de una premisa: la ciencia sin la ética no es ciencia. Afortunadamente, el Vaticano respondió de manera inmediata al llamado y logramos que el Vaticano nos diera el aval del Vaticano y que la mayoría de los científicos y de los éticos, porque siempre la Cátedra de Genética y Vida Humana a partir del año 2000, tiempo ininterrumpido de la cátedra, han llegado a Monterrey, enviados por el Vaticano a la Universidad de Monterrey, un científico y un ético. Y esto nos ha hecho que tratemos temas sumamente importantes, muy complejos de comprender, con gente muy avesada en ese campo y muy estudiada en el análisis mismo del Vaticano, de ver a quién envía y a quién enviaba a partir de ese momento para tocar temas, pues 20 años, imagínense ustedes los temas que hemos tratado, la forma como se ha conducido la cátedra y extraordinaria el mantenimiento y la permanencia de la cátedra durante 20 años con la ciencia, con la ética, para dar respuestas que la gente de una manera más accesible lo comprendan y puedan tomar decisiones más adecuadas.
La cátedra ha venido a lo largo del tiempo aportando a la comunidad conocimientos y entendimiento de lo que está pasando en el área de la genética y de la genómica. Las tendencias que están emergiendo en esta área, que es un aspecto muy importante, pero de una manera en la cual esta puede acceder a los diferentes segmentos del público y no necesariamente a una comunidad científica, aunque son expertos quienes acuden y hacen sus ponencias. Por otro lado, ha aportado conocimiento de las experiencias que han tenido quienes participan en ella como conferencistas en los campos profesionales en donde han aplicado dichos conocimientos y desarrollado el conocimiento del mismo. Recordemos que la genética ha evolucionado rápidamente en los últimos años. Y por otro lado, otra aportación importante es entender sobre todo las interacciones del ámbito y el concepto de la genética humana y de las entidades o de los seres vivos o no vivos que pueden estar teniendo un impacto sobre la persona, su salud y las realidades que está enfrentando. Los retos que ha tenido la cátedra a lo largo de este tiempo creo que han sido diversos. Voy a mencionar tres. El más reciente, la necesidad de migrar de la presencialidad hacia la virtualidad en el contexto de pandemia y de incertidumbre que estamos viviendo. El segundo reto ha sido mantener el balance apropiado entre los componentes científicos y éticos que generalmente se presentan en la cátedra a través de las diversas conferencias. Finalmente, en tercer lugar, el conjuntar a los expertos mundiales en el ámbito de la genética y en el ámbito de la ética ha sido también un reto que se ha sabido sobrellevar de una buena manera. pero debemos de enfatizar que la postura ética es acorde a los valores de la Universidad de Monterrey. En este día queremos iniciar la edición del 20 aniversario de la Cátedra de Genética y Vida Humana. Para nosotros es un gusto y esperamos que sea de mucha utilidad y aprendizaje para todos los que estamos presenciando este evento.
Ahí vamos en 3, 2, 1. Vaya.
Buenos días a todos. Mi nombre es Ana Sandoval, de la Escuela de Medicina, y en nombre de la Universidad de Monterrey les doy la bienvenida a la Cátedra de Genética y Vida Humana que está celebrando su 20 aniversario. Agradecemos la presencia de nuestros directivos e invitados especiales. Mario Páez, rector de la Universidad de Monterrey. Eduardo García Luna, vicerrector de Ciencias de la Salud. Monseñor Rogelio Cabrera, arzobispo de Monterrey. Florencia Infante de Garza, presidente y fundadora de la Cátedra de Genética y Vida Humana. Antes de iniciar, les queremos invitar a que si tienen preguntas durante la sesión, pueden escribirlas en el chat que hemos habilitado para que les demos lectura al finalizar cada conferencia. Trataremos de responder todas las preguntas que nos hagan en el margen de tiempo que hemos destinado para esta interacción. En este momento, invito a nuestro vicerrector de Ciencias de la Salud, Eduardo García Luna, para brindar unas palabras de bienvenida.
Buenos días. Buenos días. En lo que es la transición de imagen, quiero expresar que es un gusto contar hoy con la presencia de todos ustedes en un día tan especial para nuestra comunidad universitaria, al cumplir 20 años de la Cátedra de Genética y Vida Humana. No es una casualidad de que estemos con mucha alegría compartiendo esta ocasión con ustedes. Esta cátedra se ha distinguido desde sus inicios por su carácter innovador, desde la propia naturaleza de la iniciativa, que se ha mantenido vigente al abordar año tras año temas que invitan a la reflexión y al crecimiento a través de reconocidos exponentes de distintas partes del mundo. Es un evento que ha nacido para ser de todos, y para nosotros es un privilegio que la Cátedra de Genética y Vida Humana haya encontrado en la UDEM un lugar para instaurarse y para crecer. En esta edición tan especial reflexionaremos juntos desde los orígenes de COVID-19 hasta la respuesta que hemos tenido ante los desafíos que surgieron con su llegada. Estoy seguro que disfrutarán de esta edición. Muchas gracias.
Agradecemos al vicerrector García Luna por su
Paso la palabra a la doctora Soraya, por favor.
Agradecemos al vicerrector García Luna por su mensaje. Mi nombre es Soraya Mendoza, de la Escuela de Medicina. Y ahora damos la más cordial bienvenida a nuestro conferencista, el profesor Dominic Dwyer, con su conferencia Entendiendo, origen, pandemia. Y antes me permito leer una breve semblanza. El profesor Dominic Dwyer es médico biólogo especialista en enfermedades infecciosas del Hospital Wesley en la Universidad de Sydney, Australia. Dwyer es director de Tecnología de Salud Pública en el estado de Nueva Gales del Sur y también ejerce como director del Instituto de Patología Clínica e Investigación Médica. En esta misma instancia, dirige el Centro Nacional profesor Dominic Dwyer es médico virólogo especialista en enfermedades infecciosas del Hospital de Westmead en la Universidad de Sydney, Australia. Dwyer es director de Patología de Salud Pública en el estado de Nueva Gales del Sur y también ejerce como director del Instituto de
Well, thank you very much for the opportunity to present to you. It's a great honour to be asked to speak to the University of Monterrey. And I know that Mexico has been very much in the forefront of many infectious diseases issues over many years, including back in 2009 with the influenza swine pandemic. So look, I'm going to talk today about the origins of SARS-CoV-2, which is the virus that causes COVID-19, of course, as you are all aware. So I will share my screen. Okay, so I can't share my screen, so I will wait for my slides to appear on the screen. Thank you. So I'm going to talk about the origins of this virus, which, as you know, started in the very end of December 2019 in Wuhan, a large city in China. Next, please. So this is the situation we have now. As of a few days ago, we know that the world was alerted to the presence of this new severe pneumonia on the 30th of December 2019. And by now, as of a few days ago, we have had over 250 million cases of COVID-19 around the world, with over 5 million deaths. This, of course, is a great underrepresentation of the total number. It is likely that this number of both cases and deaths is many times higher. Next slide. So, when this disease appeared and started to spread around the world, the World Health Assembly requested the World Health Organization and China to investigate the origins of this virus. And this was both to understand what was happening now, but also to prepare the world better in the future if other pandemics should occur. There were two parts to this study. One was the phase 1, reviewing how the virus started in Wuhan, and reviewing some of the early evidence to see that where the origins of this virus occurred. But the second and very important part was to put in place methods to try and undertake further examination of where this virus might have emerged, not just in China, but indeed all around the world. So, next slide. So there were a number of participants selected from around the world. Next slide. The next, thank you. to show his power and strength. And this has been very much an area of controversy with the current president of Trump. Next slide, please. So this is a very important slide. This is the epidemic curve of the first 400 cases in Wuhan. And our role was to look at the cases that occurred in December, so in the beginning of that epidemic curve, the cases that occurred in December. And it was obvious that many of the cases had exposure to the wet market in Wuhan, but also the cases that were occurring early in the epidemic curve that were not associated with Wuhan. I think the other important feature here is marked with a red asterisk, and that is just to show that the viral sequence data that confirmed the new virus was available at the very end of December 2019. But in fact, it was about 10 days or so before that sequence was released to the rest of the world. And that gap is actually important because, as everybody is aware, if you have the sequence of the virus, then you can develop diagnostic tests and also understand perhaps the origins of the virus. So this delay was problematic. Next slide, please. So we looked at some of the epidemiologic features, and these relate to the mortality of pneumonia in Wuhan, in the province around Wuhan, which is called Hubei province, and indeed in the rest of the country. And the point of this is that there was a very large spike in mortality in January 2020 in Wuhan that started in Wuhan before it went to the rest of the province, and indeed before it started to spread around the world. The important thing here is that the mortality or the deaths from this infection often occur some weeks after the infection is spreading in the community. So if there was a rise in cases in January, that clearly is reflecting infection in the community in at least December beforehand. Next slide, please. And then later on in December through to January, and you can see that the cases were, and this is a map of the districts of Wuhan city, and you can see that the cases were very much clustered in one particular part of Wuhan. And as the virus spread in the community, more and more cases started to appear in the other districts of Wuhan. And the same thing occurred with the deaths, so that as deaths initially occurred in the central part of Wuhan, it's then started to spread over the next few weeks to the rest of the city and indeed to the rest of the world. Next slide, please. This picture is a picture of the Wuhan market, wet market, called the Huanan market. Next slide. And this is important because we were able to draw a map of all the stalls in the market and map where the infections were occurring in that market, both the people selling products in the market, but also attending the market to buy food and so on. And one of the features was that this is a very big market, many hundreds of stalls in the market, but it wasn't easy to find any common exposure in the market. There wasn't one particular store that seemed to start everything off. The virus started to appear in a number of locations within that market. Next slide, please. And this is, we visited the market, and these are some pictures from the market, and you can see that this is very much a good place to have an outbreak start. There are lots of small, dark, narrow corridors. We had open drainage. We have pits and cages that live animals were kept in. And on the right-hand side, you can see living quarters where people sold products on the bottom level and lived or rested in the top level. Next slide, please. We also know that there were a range of animals sold in the market. In China, lots of different animals are farmed for selling in marketplaces for meat, including animals that we might normally regard as wild animals. So all sorts of things are sold in the markets. You can see porcupines here, you can see a bear, you can see a little deer, you can see reptiles, and you can see where people live, of course. So in other words, in this market there were live animals of different types being sold, and of course we now know that there are many different animals that can be quite easily infected with this virus. Next slide, please. This is an important picture because this shows the relationship of a market with the rest of the city. So you have the market, it's crowded, it's busy, and very close by you have a very large population of people living. Most people in Wuhan live in apartment buildings, often 50 stories high. So you can see that there are many hundreds of thousands of people living in very close proximity to the marketplace. Next slide, please. The other thing we also looked at are some of the genetic sequences of the virus to try and understand the pattern or the spread of virus in the community. Now, as everybody knows, the SARS-CoV-2 in the top of this phylogenetic tree shows that the SARS-CoV-2 is very closely related to a bat coronavirus, and we do also know that there's a very large number of coronaviruses that are present in bats in the wild. And not only can they be present in bats, but they can sometimes be present in in animals that have contact with bats. And one of these that's been studied a lot is an animal called the pangolin, which is present in China and indeed in Southeast Asia. And this virus is able to infect pangolins, and pangolins have been, they get sick with this virus, but they're also sometimes traded illegally for food and other purposes. Next slide, please. From the human point of view, we looked at sequences from the very early cases of COVID-19 in Wuhan. And this is a network of the early sequences, and it shows a number of things. First of all, it shows that even in this very early period, there is already some viral genetic diversity, and that even in the cases that have a blue colour here, even in the very earliest cases from December, there was also already viruses that were slightly different from each other. And this is important because this suggests, of course, that the virus had been circulating in Wuhan for longer than what was being recognised by Chinese medical authorities. So yes, they picked up cases of severe pneumonia in hospital, but the virus had obviously already been spreading in the community for some period of time before that. Next slide, please. So one thing that was problematic, and this has been seen in other parts of the world, is that, and I won't go into the detail, but in those very early days, there were reviews of the sequences from a number of different laboratories using different sequencing protocols, using different bioinformatics or analytic protocols, meaning that it's very difficult to compare one sequence versus another. In other words, the ideal way to look at these sorts of outbreaks is to have a centralized and agreed method of doing the sequencing so that the best quality interpretation can be made. And also what is important is that the viral sequence data has to be assessed in conjunction with the epidemiologic data. And that really is terribly important. And indeed, one might say that in the early days of the outbreak in Wuhan, there wasn't a strong connection between the laboratories doing the sequencing work on the virus with the clinical and epidemiologic experts trying to understand the patterns of spread. Now we do things differently, but it just goes to show the complexity. Next slide, please. We also did some modelling, and so have many groups around the world, to using technology to try and understand or model the time to the most common recent ancestor of the virus. In other words, can you look at the sequences that have been generated now to go back to see when they started to spread in humans? And without going into the detail, if you review the evolutionary rate of spread of these viral sequences, you can estimate that the virus must have been circulating at least by the middle of November 2019, and some suggest even earlier than that. In other words, the virus was circulating for a period of time before people started to recognise what was going on. We also know that a number of countries around the world, including Spain and Italy and France, that they have had some detections of SARS-CoV-2 either in clinical cases or in blood samples or in sewage samples before December 2019. Now that doesn't mean that the virus started in those countries, but what it might reflect is the fact that the virus had been spreading not only in Wuhan, but starting to spread around the world before people realised what was going on. Next slide, please. Another area of work was around the animal and environmental studies, because could we do work on the animals being sold in the market or the animals being sold in other parts of the country, not just live animals, but also butchered animals or frozen meat, all of those sorts of products that might be sold in the market. We know that SARS-CoV-2 can be seen in lots of different animals. A lot of investigation was done of bats, for example, in Hubei province, other wildlife and domestic animals. All of these studies, of course, were negative in detecting SARS-CoV-2, but of course the trouble is that this investigation occurs well after the virus might have moved from animals into humans. So just because you find everything negative today doesn't mean that animals weren't positive some weeks or even months beforehand. We also know that the virus will survive for reasonable periods of time in the environment, including on frozen food, packaging, cold chain products and so on. And of course, these have been investigated, but by the time these investigations are done, it's often distant to the period of time when samples might have been contaminated and are sold. And also environmental testing in the market showed that there was lots of virus spread around the market, but that doesn't help tell us whether it's due to the people that were infected in the market or animals that may have been infected in the market. Next slide, please. The discussion, of course, of the origins of these viruses means that we have to go back and look at viruses in bats and wild animals, not just in China or in southern China, but indeed in other parts of Southeast Asia. And we already know that there have been related viruses, not the direct parent of SARS-CoV-2, but related viruses in bats in various countries, in Thailand, in Laos, in Vietnam, and indeed coronaviruses have been found in bats in many countries around the world, and so on. It's also been found in some animals, as I mentioned before, the pangolins, of course, which are illegal in China and many other countries, but appear to be being sold for various food and other purposes. And this is important because some of the strains in the pangolin are actually quite closely related to SARS-CoV-2. Next slide, please. The other thing is that we do have to remember that these the bats, for example, don't live in just one country. They can cross borders very easily. And indeed, the distribution of the various bats and so on that are positive for SARS-CoV-2, in fact, is not just in China and southern China, but also elsewhere in Southeast Asia. And indeed, the geography of these bats can be quite extensive, going right across Central Asia and indeed almost into Europe. So in other words, that's important because we can't just limit any studies to China, where the first clinical cases in humans appeared. We also have to look at the animals in a much wider environment, and this brings the whole idea of One Health, of animal and human working together to try and understand the origins of disease. Next slide, please. So this is, and the next slide as well, please. These slides really just show the extensive amount of testing done on animals in the market. Next slide, please. And also elsewhere in China. And the results of all of these studies have been negative to date. But as I said before, looking for these animals may have missed the time when the virus came through those animals into humans. So the fact that they're negative is not an exclusion that some animals may have acted as some sort of intermediate host in between bats and humans. Next slide, please. The other important thing I'd just like to emphasise to this audience is the impact that this outbreak had on the doctors and the hospitals and the nurses and the medical system in general in Wuhan in these very early days. So we spoke to some of the first patients infected who survived with COVID-19, and we also talked to the doctors at some of the hospitals and the community centres. And I think that what we have to understand how difficult it was for people to work in that environment in the very early phases, because they didn't know what the virus was, they did not have any diagnostic tests, they did not have any treatments that they could use. Obviously there wasn't a vaccine. So the pressure on the medical system was very important. We also spoke to some of the communities that were very affected in the very beginning of COVID-19 in Wuhan. And if I can just tell one quick story, I spoke to a doctor who was an orthopaedic surgeon who told me that his wife was an internal medicine physician in one of the hospitals. She was looking after some of the very early COVID cases. She herself got COVID-19 and died of COVID-19. So this doctor was now left without his wife and having to look after a 2-year-old child. And so I just tell that story because at the end of all of these discussions about the number of cases, the origins of the virus, what people should do or shouldn't do, new vaccine, at the end of the day, there are people with this disease, and we mustn't ever forget the personal impact on people with this disease. Next slide, please. The other area that's had a lot of media attention has been the Wuhan Institute of Virology, which is a very well-known research institute in China and well known around the world. And this has been the subject of a lot of discussion, often in the newspapers, and sometimes used for political purposes and point scoring and so on. So the Wuhan Institute of Virology does do research on bats. They publish extensively. They have identified many bat coronaviruses. Next slide, please. Including one virus that is most closely related to SARS-CoV-2 called RATG13. And I mention that particular virus because people have suggested, well, perhaps there was a leak from the laboratory, perhaps it was deliberate and deliberately created. And they often go back to this RATG bat coronavirus. But the important thing is here that bat coronavirus we have a genetic sequence from the virus, but we do not have any live culture of the virus. Not all the bat coronaviruses are easy to grow in the lab. In fact, they're often quite hard. So this virus that's talked about as being the possible source of a laboratory leak, in fact, has never been cultured in the lab. And to get a laboratory leak, generally, you need to be culturing or growing the virus in the lab. So I think, we examined the idea of could this be a laboratory leak, or could it have been an accident, or indeed even deliberate? And I think we need to understand how laboratory leaks happen. They can be an accident when collecting the sample, when handling the sample in the lab. But more importantly, they usually occur when you're growing the virus up in the lab for research purposes or studies or what have you. People have suggested, including politicians around the world, that perhaps this was done deliberately in China. And yes, you can have deliberate release of viruses and so on, and terrorism and so on. This has happened occasionally in the past. But for this to happen, you have to actually have a complete cover-up in the country to say that they are doing this work. And there's been no evidence of this anyway. And even the United States intelligence agencies have gone and done their own investigation and have not found any conclusive evidence. So to say that there has been a laboratory leak or a deliberate release of the virus is an easy thing to say, but in fact, you need evidence to take that further. We can also make the point, in fact, that SARS-CoV-2 is not a very good biowarfare agent. Why would anyone choose that virus to attack another country? Because it doesn't kill everybody. It generally kills the older people or whatever. It doesn't kill people of a younger age generally. So even if you wanted to propose that this was a biologic agent, it's actually not a very sensible choice. Next slide. Next slide. Thank you. So in the end, we presented and examined a number of different hypotheses as to what might be the origin of the virus. And you must remember that none of these steps are simple. They're often quite complex. So if you look on the left-hand side, you might have had a bat or some sort of animal that had the virus, or an animal infected by a bat, and that virus or animal can go into a human, or it can go into an animal, or it can go into products of animals. And of course, as we well know, once the virus gets into either humans or animals, it can undergo its own genetic evolution to become either more virulent or less virulent, and so on. And we see that, of course, now with the different strains or variants of SARS-CoV-2, such as the Delta strain, where the virus has become more transmissible after it circulates in humans for a while. The other thing you have to add in is human behaviour, mass gatherings, for example, be they sporting events, religious events, marketplaces. Those sorts of activities also promote the spread and transmission of virus around the world. So what we were doing was really looking at these different transmissions and trying to put in the evidence in the different pathways to come up with a story. Next, please. Next slide. So this is just one example where marked in red as to how we work through this process. So if you take a virus that's come out of a bat and you say that it's come into animals, it may then spread directly to the human above, or it may spread amongst the other animals, and while spreading in animals, gain some of the molecular features that in turn make it more easy to spread into humans. If it gets into animals or humans, it can of course get into the markets, it can get into mass gatherings, it can get into laboratories and so on. So you actually need a whole series of events to occur for the virus to take off in humans. We actually now know that there's a lot of viruses that come across from, for example, bats into humans, but they don't go on and cause disease or spread. And I think that's important. These sort of accidents or changes probably happen very commonly, more commonly than we think, but only very occasionally, fortunately, do they go on to cause and spread disease. Next slide, please. So in the end, we have a number of hypotheses for the origins of SARS-CoV-2. And the WHO mission was designed to try and work through the different hypotheses and try and say which were the more likely and which were the least likely. And based on the evidence we have to date, it's most likely that this has come out of an animal such as a bat and probably gone through some sort of intermediate animal into humans. The other things, such as spread through frozen products or escape from a laboratory, are still on the table as a hypothesis to examine, but hard to take forward unless you get more evidence to prove that. So you really need to be driven by the scientific evidence rather than hearsay or speculation. The other issue is, did it start in China and spread to the rest of the world, or did it start somewhere else and then got to China before it spread to the rest of the world? Now this becomes very political, so of course some countries will say, oh well, it all started in China and spread to the world, it's China's fault. But of course the Chinese might say, oh well, it could have started in a bat in Vietnam or Thailand or somewhere, then come into China, into Wuhan, and then spread. And to be honest, this is not necessarily about examining the evidence, but trying to perhaps blame other people other than themselves for where the origins of the virus were. And that's a very hard discussion to have. Next slide, please. So this, as I mentioned in the beginning, the second thing was to come up with studies to take this further forward. And I'm not going to go into the detail, but there were a range of recommendations in the report based on reviewing some of the data in more depth, including some of the data on the very early cases, going back and looking at animal farms and people who work in animal farms, going back and looking at people who work or who have close contact with bats in various different countries, and so on, a whole range of different things. And these are all important studies to do. Next, please. So what has happened since this mission to China in the beginning of 2021? Next slide. So there's some important papers that have come out to further support the idea that this is of animal origin. So there was a paper that came out after the WHO mission from Chinese and Canadian investigators. Next slide, please. And they showed that in fact there were a range of animals being sold in that particular market in Wuhan over some years before the outbreak occurred. And this is some of the pictures that they took, a whole range of different animals of one form or another. And this is important because the people had said, oh, well, there are no animals in the marketplace that couldn't possibly have started there. But in fact, there is evidence, clear evidence to show that animals were infected, there, were present there, and that those same animals themselves can be infected with SARS-CoV-2. Next slide, please. The other thing is that there's more and more papers coming out, and I'm not going to go through them in any detail, but I just want to say that a number of people have said it must have been a laboratory leak because some of the genetic features of the virus are very much targeted towards humans. And these include things like where the virus binds, the specific receptor on human cells called the ACE2 receptor. There's also what we call furin cleavage sites, where the virus is cleaved by human proteins to start its replication process. Anyway, the point of all of these papers are that all of the features in the genome of the virus that cause tropism or the ability to infect him are present in viruses, in bats, in the wild. So you don't need to propose, oh, well, somebody must have done some genetic engineering of the virus in a research lab. All of these features are present naturally in the wild, in bats throughout Southeast Asia. Next slide, please. Then there have been a number of reviews around some of the sequences. People had postulated that the Chinese had some sequences of the virus, or put them up initially, took them down and so on. These have all been reviewed, and the sequences are in fact available. And people have also independently gone over the WHO, World Health Organization report, to confirm the validity of the data. And again, these all show that the original conclusions from the WHO report are in fact quite accurate. Next slide, please. The other, I think, important and distressing part of all of this work is that politics has been used to attack the science and attack the WHO and attack other people because it doesn't suit political agendas. As I said before, countries to perhaps not talk about their own problems with handling the pandemic, like to attack other countries because it's a way of deflecting attention to their own shortcomings. And so there's been a lot of theories, including conspiracy theories, in many countries around the world, including in the United States and North America, and in Europe, Australia even, where people have attacked by saying, oh, well, it's the Chinese have done this deliberately in their lab, and those are not based on evidence, but based on a political agenda. And I think it's been unfortunate that trying to do science, which is of course evidence-based, is complicated by non-scientists attacking the data without understanding the data themselves. Next slide, please. So where are we now? So we wrote a paper in the journal Nature on the left-hand side there saying that the longer the world takes to go back and do the next phase of studies, the harder it is to go and do the answers, get the answers. So because of all of the political fighting between countries and so on, we're now nearly 2 years after the pandemic, and the WHO hasn't gone back to do further work in China. You can also say that the WHO and the President have now formed a new group to review SARS-CoV-2, but also to prepare the world for approaches to to understanding the next pandemic when that occurs. And I'm sure there will be one somewhere along the line. But I think the other important thing in the top right is that there was, while we were away in Wuhan, we knew that President Biden in the US was commissioning his intelligence agencies to see if they had any evidence of a laboratory leak and so on. Now, without going into the detail, essentially there's been no evidence found of a laboratory leak or deliberate release of this organism as a hypothesis for the beginning of the outbreak. Now, that we still leave that idea there, but there's no evidence. So unless further evidence comes, or any evidence comes forward, one can really take it further. Next slide, please. So this is my last slide. So I just wanted to say in conclusion that origins of outbreaks are very complex and they may take some years to answer. We know that, for example, with SARS back in 2003, it was about 14 or 15 years before the animal origin of that virus was found. We know that with Ebola virus in Africa, that we've had outbreaks in Africa for maybe 40 years of Ebola, and it's really only in the last few years that the origin of that virus has been identified. The important point is here that it is naive to think that we can come up with origins of viruses and pandemics easily. It often takes time. To do that, you have to have collaboration, transparency, openness, and freedom from political agendas to do the research to answer the questions. The consensus, general consensus, or the most common consensus, is that an animal is responsible for this pandemic, most likely from bats, and there's a lot of work that needs to be done in that area, and we also need to understand that again, this requires openness and sharing, and that this work can't just be limited to China, but indeed has to happen in all parts of the world, including parts near China. And science is the key. Politics makes it more complicated. The media approach to politics and science is also complicated and can make things difficult. And we have to try and get away from complicating the science, the good science, with all of these other factors, so that we can answer the questions that will then give us the answers about this pandemic, but also protect mankind in the future from other pandemics. You can have a reference there at the end. You can read this report if you like and make your own conclusions, and many people have done that. So look, I'll finish up there, and again, thank you very much for inviting me to talk to you. It's a great honour, and I do hope that next time, once COVID is finished, that we can meet face to face rather than doing things over Zoom. So thank you very much for the invitation and for listening to me today. Thank you.
Agradecemos al profesor Dominic Dwyer por su interesante conferencia. A continuación, daré lectura a algunas de las preguntas que recibimos durante la plática. Esta pregunta la realiza el doctor Eduardo García Luna. ¿Qué expectativa se tiene en la evolución del virus? ¿Hay mutaciones que van reconfigurando el virus? ¿Y qué expectativa se tiene en torno a su evolución?
Well, I think that's a very important question. The history with RNA viruses like SARS-CoV-2 or influenza or HIV or hepatitis C is that they mutate over time. And they mutate for a number of reasons. They mutate just because RNA viruses mutate anyway, but they can also mutate under immunological pressure, and that can include the use of vaccines, for example, and it can include the use of drugs and treatments and so on. We've already seen quite some evolution of different variants of SARS-CoV-2, and as we discussed earlier, the Delta variant is the one that now seems to be the most contagious, the most transmissible. So I think we will continue to see evolution and mutations appearing. The question one asks then, are those mutations important? Many of the mutations are not important, but some of them, for example, Delta, are important. And I guess what we have to do is surveillance of the genetics of the viruses to make sure that we are not seeing viruses emerging that are escaping from vaccination and treatment. Thank you.
Gracias. La siguiente pregunta la realiza Ana Karen Salinas. Si no hubiese cambio en la venta de animales, ¿qué tan pronto podría presentarse una nueva epidemia como COVID-19? ¿Cree que esto tenga un impacto cultural en China?
Very much so. So the use of wild animals and the often the slaughtering of wild animals in marketplaces is a feature in many countries around the world, in China and Southeast Asia and in Africa and so on, in the Americas. So it is the key is our connection as humans with animals. And so I think one of the difficulties with wild animal markets or animal markets is that they're often poorly controlled. So that, of course, the meat that we buy at the supermarket, for example, is very well controlled, the quality checks and microbiological checks and so on. But going to some of these markets and buying an animal, slaughtering it there, taking home some of the meat, that sort of thing is a recipe for outbreaks, not just of viruses, but bacteria and other things as well. So I think as humans, we have to look at how we interact with animals that we use for food or have contact with. And I think we have to have regulation about such animals. In principle, many of these wild animals are illegal, but of course that doesn't stop them being sold illegally. So I think there's a lot of work to be done about changing the regulation of markets, but also the cultural practices that promote this sort of process. So again, I think China and indeed other countries have to seriously look at the way animal markets work and what might be alternatives that are safer. And one of the difficulties is that in China, for example, the wild animal market is a very big industry that employs many, many hundreds of, indeed millions of people. And so the economic impact of making these changes also needs to be considered.
Muchas gracias, doctor. La siguiente pregunta la realiza el doctor Jorge Luis García. ¿Cómo esperaríamos que la prevalencia y la severidad de la enfermedad cambien con la transición hacia las zonas endémicas? La siguiente pregunta por parte del doctor Jorge Luis García es la siguiente. ¿Cómo esperaríamos que la prevalencia y la severidad de la enfermedad cambien con la transición hacia zonas endémicas?
Well, I think this is one of the difficulties, isn't it? Of what happens to viruses when they first enter the human population can change over time. So sometimes viruses can be very severe in the beginning in humans as they jump from animals into humans, but as time goes on, the virus evolves to cohabitate with humans. So one expectation might be that, well, look, things were very bad in the beginning in terms of death and disease in the beginning, but as time has gone on, has the virus become less severe? I'm not sure that we have that evidence with SARS-CoV-2, and the new strains that have emerged, such as Delta strain, have caused problems because they are even more contagious, more transmissible. So I think we're still in the early stages of the interaction between this virus and humans in terms of what sort of disease it might occur. We do know historically that things like influenza, like the pandemics of influenza, or indeed the very early coronaviruses that we now, that are in humans that cause, for example, the common cold, that those viruses are not terribly severe. So it may be that this virus becomes less severe over time. We really don't know at this stage. So I think, again, this becomes an ongoing area of study to look at the viral genetics, look at the clinical aspects, look at the responses to vaccination. All of those sorts of things together are really what need to be observed to see whether the virus is going to change. I personally think that as time goes on, this virus may become less important to humans. It may become another common cold virus, but that is purely speculation at this stage. Thank you.
La Universidad de Monterrey, a través de la Cátedra de Genética y Vida Humana, agradece su valiosa participación. Como nuestro segundo invitado magistral, le damos la más cordial bienvenida a nuestro conferencista, el Dr. Hongshou Liu, con su charla Exploring the Viral Host Factors Related to COVID-19, la cual verán a continuación.
Hello, distinguished
afiliado a la Universidad del Sur de Ciencia y Tecnología. También ejerce como codirector del Centro Clínico de Salud Pública de Shanghái en la Universidad de Fudan. El Dr. Hongshou Liu es decano asociado del Hospital Huashan de la Universidad de Fudan y presidente de la Sociedad de Medicina Tropical y Parasitología de la Asociación Médica China. Cuenta con reconocimientos como the Yangtze River Scholar, National Thousand Talents Program, the People of the Year Awards 2016 y Shanghai Science and Technology Award. El Dr. Hongshou Liu es codirector del Centro de Colaboración para el Manejo Clínico, Capacitación e Investigación de Enfermedades Infecciosas Emergentes y Reemergentes de la Organización Mundial de la Salud. Ha publicado más de 40 artículos sobre enfermedades infecciosas. Recibamos al Dr. Hongshou Liu.
Hello, distinguished professors from overseas and from other countries. This is Dr. Liu from the Shanghai Public Health Clinical Center and from Shenzhen Third Hospital. So I have two hospitals. Before August, last August, I worked in Shanghai Public Health Clinical Center. Then I moved to Shenzhen, the third hospital. The third people's hospital is also the designated hospital for any kind of infectious disease like COVID-19, H7N9, influenza, HIV, AIDS, and tuberculosis and hepatitis. Now I'm working in the Shenzhen third hospital, and in Shenzhen, this is the only hospital for COVID-19 cases. So today, I would like to report my research on the exploration of the viral host factor related to the patients, and also some novel treatments for this kind of cases. So both of the hospital they have a lot of COVID-19 cases, so I would report the clinical research about COVID-19 in both hospitals. Majority is my research. And the COVID-19 pandemic in the world will still continue for many years, but the COVID-19 will change the human being, and it caused a lot of disaster, and lots of patients have died of this. This is the WHO report. The epidemic is still keep going, and you can see every day 500,000 patients who are infected, and nearly 7,000 of the cases they died of the disease. So it is still in a very high level of the mortality and morbidity in the world. And this is the mutation variety of the virus. Alpha, beta, delta, gamma, and some other mutation subtype or the new variety of the virus will still emerge in the future. And some of the animals, especially for the wild animals, when they infected the virus can mutate to suitable for the new animals. And this is a challenge because the new animal, when they infected the virus will mutate a lot. So the mutated virus then can feed back to infect the human being again. And for the human being, this kind of variety subtype is a novel subtype. So they can escape the vaccine response. And so we should surveillance this kind of mutation. We should surveillance and stop the new virus subtype from the animals to human being. And you can see now in the world, even some kind of country like China, like in Europe, they had a lot of people who have already have 2 or 3 shots of the vaccination, but there are still a lot of patients who are infected. So there are more than 300 of vaccine clinical trial, phase 1, phase 2, phase 3, and more than 20 of vaccination has already been permitted for the human being vaccination. But all those vaccination, all those kind of, including mRNA and some other vaccine, they cannot prevent the virus infection. Even they can prevent the patients hospitalized. They can prevent the patients become very serious, but they still all those vaccine, they do not prevent the virus infection. And the patients who are infected, the patients who have already been vaccinated, even they have a high level of the neutral antibody, the patients can still be infected, and the patients can also still infect the virus into others, can transmit the virus into others. So we should have some kind of vaccine. It is a kind of respiratory protection, and this is the Nebula virus, Nebula vaccination. If we use the Nebula and we have the antibody, we have the response in our respiratory, then we can prevent the virus infection. So in the future, we should have some kind of vaccine. It can be respiratory response, and also have the shots, and then we can have the T cell response. We can also have the the response of the we can use the antibody to neutralize any kind of infection. At the beginning, WHO, they have a lot of awareness to the world, but the world, especially the American, especially the Europe, they do not listen to WHO. And at the beginning, when Wuhan had the epidemic, and our country under the leadership of President Xi, we respond so quickly, and we control the pandemic. And each of the patients, they get a good treatment. And we have all the contact of the cases quarantine. And we even quarantine the whole city, the Wuhan city. But you can see, even the Wuhan city, they have 20 million of the citizens. But the whole nation, we contribute a lot. Some other cities, we have our own medical staff. We come to Wuhan to help the local people. Because in Wuhan, the local doctors, the nurses, they may have already been infected. And some of them even can transmit the virus into others. So all the doctors and nurses, they quarantine themselves. They do not to see patients. Then we have our doctors from other city. We treat all the cases. We quarantine, and we have the infrastructure in China. In one of the city, they have the problem. They cannot afford. They cannot treat all cases. They do not have enough staff, and then the other cities, the other provinces, they can be helped. So this is the Socialist Party. This is our China. Now we can have this very good infrastructure. So this is why we use very little time. Within 2 months, we control the disease. We control the COVID-19 in China. And last year, since May, majority of the cases, they are imported cases from other countries. And we have several of this kind of imported cases, but we detect the patients in the early stage. And then we report, we quarantine, and we treat all the cases. So if we find some of the important cases, if we find some of the epidemic or sporadic cases, and we quarantine all the patients who contacted or subcontacted with the patients, and we can also use the big data to find all the cases who contacted, who may contact, who may be potential infected cases. So we use the big data to find all the cases, and then we quarantine. And then we have the PCR test, or use the nucleic acid to find all the positive cases. Even they are asymptomatic, even they do not have any kind of signs of the disease, but we can find all the cases, and we quarantine, and then we can control this kind of sporadic or epidemic of the cases. So talking about the COVID-19 sequence, and as you know, in Shanghai Public Health Clinical Center, in my hospital, we have a team, the scientists. They have a lot of identification of novel virus. The team is Professor Zhang Yongzhen, and they have the first identification, and because in Wuhan, some of the patients, they got a fever of unknown origin. They will have the samples sent to our hospital to Professor Zhang Yongzhen's P3 laboratory. And in their P3 laboratory, they find the novel cases, the novel sequence, and then we report to the world at the first time. So because we have the first sequence reported released to the world, and the world can use the sequence to have the kit, and also they use the sequence to develop the mRNA vaccine. So we contribute a lot to the world. So this is the publication described how the team they report the novel virus sequence. And in China, we have a system. The system is the CDC system for the fever of unknown origin, or the fever of respiratory cases, and all the samples, they depend on what kind of bio safety we should follow. So all the samples they send to our laboratory, and also depending on the biosafety rule, we depend on the law. So we contribute a lot for the earlier finding of the virus. WHO also said they got the sequence in June 11. So this is the time we release the virus sequence. And after we have the sequence released to the world, Pfizer, Moderna, and many kind of some other company, they use the sequence for the mRNA vaccine. And a lot of company, they can have the diagnostic kit, because PCR they can use to have the diagnostic kit. So also in China and in the world, I have already have some of the conversation with them, and they also express their thanks to our hospital, because we have the first sequence. We also have the identification of the first sequence. So where is the origin of the virus? We all know the virus is from animal to human being. So where is the origin? So it is really a scientific question, because in Wuhan, we report in Wuhan there are the epidemic, but Wuhan is not the origin, because we have some of the scientific research. We find in Wuhan, the seafood market, there are a lot of some kind of seafood and animal. They from overseas, they come to Wuhan. And Wuhan is the place we first report the outbreak. But now we report it's not the origin. Maybe some of the virus, some kind of virus, they from the other country. Maybe seafood, maybe some other animals, they come to Wuhan seafood market, then they can cause the outbreak in Wuhan. Now, there are a lot of reports about some other animals, they can also be infected the virus. And the different animals, they have their own different immune response to the new virus. Then the virus in different animals may be mutated a lot to suitable for the wild animals. Now we can see gorilla, dog, cat, lion, mink, forage, mouse, and many other animals. They may have some of the symptoms when they are infected. But the majority of the animals, they do not have any kind of symptom signs. But the virus can mutate a lot. This kind of mutated virus maybe is a subtype or subset of the virus. Maybe the virus can infect human being again. So this is really a challenge. So in the future, the vaccine should have some kind of new concept for any kind of mutation. So we should have a kind of vaccine they can cover, they can respond to all kind of mutation, and they can also be response to the novel mutation of the virus in the future. So we really have a lot of challenge in the vaccination. So talk about the vaccination coverage. Nowadays, there are more than 20 kind of vaccine being permitted in different countries, like the Oxford, Pfizer, Moderna, Sinopharm, and also Sinovac. And in the world, you can see a lot of countries, they have different branch and different company. The vaccine is different globally. And you can, through the viral vaccine coverage, you know, some of the countries, they have already have the vaccine. The dosage is very large. Even there are a lot of vaccination. The whole world is still under epidemic. But in some countries, because they have the mRNA vaccination, after the vaccination, the patients who are very mild, they do not become very serious condition. They do not need to go to the hospital. They do not need to develop into very serious, do not go to the ICU. So the mortality should be low. But the patients who do not have the vaccination, they can become very serious or die of the disease. We still support all the people, if they can have any kind of vaccine available, they should have the third vaccination. Now, I would like to report my research when I working in the two hospitals. My team, and we have the dynamic of the neutralized antibody to respond to the virus, and we have more than 175 of the cases. We have the serious sample, and we find the neutralized antibody in the patients when they recovered from the disease. And we can see the neutral antibody will be decreased very rapidly. And we also have some of the pseudovirus to see if they can have the response. And we find the antibody decreased very rapidly. So that it means when the patients they get recovered, and because the neutral antibody is very low, maybe they can be infected again. So the second infection or third infection can also be happened. And 80% of the cases, they do not have the neutral antibody. So this is one of our publication. And WHO also used our finding, and they said unclear if recovered coronavirus patients are immune to the second infection. Actually, there are some of the cases, sporadic cases, they got the second infection or even third infection. That is, means maybe even we have a vaccination, we have a good, very responsible, effective vaccination is so important. So in the future, we should have a new concept to have our vaccine developed. So this is the publication. They said our research is a highlight in the past year. This is one of my team. We doing some of the sequence from the patients, and the patients they are in the early stage when Wuhan have the outbreak. They are infected. Some of the cases they come to from Wuhan to Shanghai, and then they may infect their colleague, or they may infect their family. So in the early stage, we have all the cases. We identified the virus, and then we sequence all the isolates. And we find in the early stage, the patients, they are related to the Wuhan seafood market. The virus have already been have the two clades, and they do not unify the virus. They have already developed two clades. So if the Wuhan city the outbreak, but maybe they have two origin. The two origin is not in Wuhan. It is from the other country. So other kind of origin virus, they come to Wuhan, and then they become infected first in Wuhan. We find the outbreak, and this is my publication, and this publication was reported in May 20th. We reported by the Nature, the journal. So we contribute a lot because we are the first scientific report, and we contribute a lot for the origin of the virus. And we also find if the patients, the CD4 and the CD8 become decreased, and the patients will become very serious. So for the mild or the patients do not have any very serious condition, and the CD4 and the CD8 is higher. So the CD4 and the CD8 count can predict the patients will become serious or not. And we also find the cytokine like interleukin 6 and interleukin 8. If this kind of cytokine increased and also predict the patients will become very, very serious condition. So talking about the host factor regarding to the T cell receptor, we also find in different cases they have the different situation. So we through the research we find the T cell response is so important for the cases. If the T cell response in the patients after follow up for half a year or 10 years or 1 year, we still find the T cell response to the virus, or even to some of the mutation virus. And we have the pseudo virus, and we doing some research. So we got the conclusion: T cell response is so important for the prevention of the virus, even the mutation virus. My team have the multi-center the clinical trial, including the patients from Italy and the patients from France, we find the olfactory and gustatory dysfunction in all the cases, about 41 of the cases who have the olfactory and who have the gustatory dysfunction. It may be the first symptom and the first sign of the cases. Even some of the patients, they do not have very serious condition, and they still have this kind of olfactory or gastrophagy. If the patients, they can recover, and other dysfunction can become also recovered. So we have the finding, we find the patients, if they become very serious, it should be at the second week of the clinical progress. Majority of patients, at the second week, they will become recovered. But a few of the cases, about 10% or 15% of the cases, they progressed very serious at the second week. In Wuhan and in Shanghai, there are several of the patients who are very serious. So what's reason? What's the clinical future? What's the clinical feature? And what's the immune status of all the serious cases? So we have the multiple study about all the serious cases, why they can become very serious, and we also have the publication. My team have the publication in cell research about the patients when they are infected in the latent stage, and some of the patients, they have the asymptomatic. Some of the patients in the stage, the pre-symptomatic, and we find the lymphocyte is different. So this publication again focus on the T cell response to the virus. It played a very important role in the infection, and this is different. All the cases we have the host and the T cell response to the virus, and it can also be very important for the T cell research. So in the future, what kind of treatment for the disease? Because some of the patients, they still died of the virus every day. So what kind of treatment for the patients? In China, we have a lot of Chinese medicine. For example, we find one kind of Chinese medicine. It's called the Shufu Jiezhu. This kind of herbs can be used for the treatment, and it can inhibit the cytokine storm. So it plays a very important role in the treatment, especially for some serious cases, and we have the publication. Do you know the molnupiravir is an oral drug for the treatment. They have the clinical trial, and they can also publication. So generic drugs in China, we can also have this kind of clinical trial in my hospital very soon. Some other drugs like the anti-inflammation treatments, some of the drugs like the JAK inhibitor, it can also play a very important role in the future. So corticosteroids in China can also be used in the patients. They become very serious at the second week. So we do have this kind of clinical trial. So the nasal high flow is also used in all cases when they need it. there are some other treatments. For example, the antibody to neutralize the S protein is so important. And we have a lot of clinical trials to use the antibody to treat the patients, especially in the early stage. This is the publication in Nature Communication, and it contributes a lot by the Shenzhen, the third hospital. And my research about the kind of neutralized antibody, I also find in the patients, if we use the early, especially the patients do not develop a very serious condition. The patients do not become multiple organ failure. We use the neutral antibody in the early stage. They can play a very important role. They can prevent the patients become very serious. So this is publication by the Shenzhen, the third hospital. They doing a lot, and this is one of my team. We also use the neutral antibody, and the safety and tolerability is so kind, and we also publication, and we have a lot of some other drugs, especially for the neutral antibody, and it should be play a very important role to treat all the cases. So because of my team contributor to the scientific research to prevent the disease, to treat the disease. So in France, the France National Academy of Medicine gave me the award about my contribution, and it is the first Chinese person to get this award. The award is the first award about the COVID-19 research. And in the future, we should doing a lot on the research about the very effective vaccine. So the vaccine should be a long-term vaccine, long-effective vaccine. And for my acknowledge, we should thanks to the Shanghai Public Health Clinical Center and also the Shenzhen Third Hospital and some other colleagues. They contribute a lot in my research. So thanks a lot.
La Universidad de Monterrey, a través de la Cátedra de Genética y Vida Humana, agradece su valiosa participación. A continuación, como participación especial, tenemos a Monseñor Rogelio Herrera López, arzobispo de Monterrey, y me permito leer una breve semblanza. Monseñor Rogelio es el 12º arzobispo de Monterrey. Es miembro de la Pontificia Comisión para América Latina. Monseñor cuenta con estudios en Humanidades, Filosofía y parte de Teología en el Seminario de Querétaro. Terminó su formación teológica en Roma en la Universidad Pontificia Gregoriana y obtuvo la licenciatura de Sagrada Escritura en el Pontificio Instituto Bíblico en Roma. Ahora invito a Mario Páez González, rector de la Universidad de Monterrey, a tomar la palabra.
Estimado señor arzobispo, en este momento tan
de la universidad, al señor Mario Páez, siempre con mucho interés en participar en todas las actividades que desarrolla nuestra Universidad de Monterrey, especialmente en estas actividades que acompañan el caminar ordinario de la universidad, las cátedras, que son encuentros especiales de profundización. Esta cátedra de vida humana y genética, como la cátedra de arte sacro, revisten a la universidad de una mirada muy importante que la conectan no solamente con el entorno de nuestro estado, de nuestra ciudad, sino con el entorno universal. Porque hablar de la vida y hablar del arte es hablar siempre de un patrimonio de la humanidad. En esta ocasión, algo muy importante es el tema de la vida, y que han querido en esta ocasión verlo desde la gran dificultad que ha presentado la pandemia. La humanidad, para cuidar la vida que siempre está en riesgo, tiene tres circunstancias siempre dolorosas. La primera es la de la guerra, la segunda, la de la peste, y la tercera, de la hambruna. Tres realidades que ponen en riesgo la vida humana, y no solo la vida humana, sino la vida en general, también la vida de los animales y desde luego también la vida de la naturaleza, de los árboles, de la creación. De allí la importancia que tiene esta cátedra de genética y vida humana, porque nos permite mirar este bien divino, este bien humano, este bien universal que es la vida, mirarlo en toda su amplitud, mirarlo siempre como ese don que Dios nos ha regalado y siempre con la actitud de agradecimiento a Dios por ella y también, desde luego, la acción o la responsabilidad que asumimos. Porque todo don, como es el don de la vida, primero se agradece en una actitud benevolente con Dios, pero también se convierte en una gran responsabilidad. Nos deja muchas tareas en diferentes ángulos de la vida. Tenemos que cuidar la naturaleza inanimada, la naturaleza animada, donde están los animales, las plantas, los bosques, la selva, pero de modo muy especial el cuidado de la vida humana, que está en el centro de toda la creación. Cuando Dios creó al hombre y la mujer, Él consideró que era el momento máximo de su creación. Al terminar de crear al hombre y a la mujer, dijo, todo está muy bien. De las demás realidades había dicho, está bien el mundo animal, está bien el mundo inanimado, está bien esta casa común para el ser humano. Pero cuando creó a Adán y Eva, al hombre y a la mujer, el Señor mismo quedó maravillado de su propio milagro, porque dijo, y todo estuvo muy bien. De ahí que la Universidad de Monterrey, a través de esta cátedra de genética y vida humana, quiere cumplir con esas tres tareas que tiene frente a la vida. Primero, de ver en ella un don que no nos pertenece en su origen, sino que viene de Dios. También obliga a fomentar las actitudes de agradecimiento y cuidado de la vida y desde luego se convierte en una responsabilidad que nos obliga a actuar. De ahí que tiene muchos aspectos importantes el tema de la vida. Y hemos querido en esta ocasión abordarlo desde el tema de la pandemia. Como dije al principio, hay tres situaciones que ponen en riesgo la vida humana, como la vida de todos los seres animados e inanimados. Pero referidos a la vida humana, lo que pone en riesgo es la guerra, el hambre y la peste. Y estamos viviendo esta realidad tan complicada que ha sido la pandemia, que ha tenido alcance universal, ha llegado a todas partes, ha sacudido a la humanidad, nos ha puesto en grandes preguntas, en grande desconcierto. Y en este asunto el Papa Francisco nos dio una clave para mirar la pandemia. Primero, no es un castigo de Dios. La pandemia no es un castigo de Dios. El Señor quiso, después del diluvio universal, nunca castigar a los hombres ni con pandemias, ni con guerras, ni con diluvios. Simplemente en ellas hay una resultante de responsabilidad humana, pero también hay algo que nosotros no siquiera podemos ubicar sus causas. Se discute este virus COVID-19, dónde tuvo su origen, y empiezan las discusiones, pero siempre está el misterio que encierra siempre la pandemia y toda peste y toda problemática que abarca la humanidad. La pandemia, pues, nos ha puesto a todos en la conciencia universal. Fíjense bien cómo el Papa nos preparó proféticamente para este acontecimiento. Poco antes de que viniera COVID-19, el Papa nos invitó en su exhortación Fratelli Tutti, todos hermanos, a tomar conciencia de que ni la religión, ni las opciones políticas, ni los estados financieros pueden ser la razón de división entre los seres humanos, que todos estamos llamados a construir fraternalmente el mundo, a vivir la amistad social. Eso preparó el ánimo, al menos del pueblo católico, para mirar la pandemia en su tragedia y en su dolor, pero nunca mirarla como un castigo de Dios, sino siempre como un kairos, como una oportunidad que el Señor nos da para vivir la generosidad y la caridad. Todos los problemas que tiene la humanidad, como la guerra, como la peste, como el hambre, son para nosotros oportunidad de generosidad. Es movernos, el Señor nos mueve a vivir la caridad. Y así hay que mirar esta pandemia en medio de sus trágicas consecuencias, de tanto llanto y dolor que hemos sufrido y que todos de alguna manera u otra hemos tenido pérdidas. Pero es muy importante mirarla siempre como una ocasión de recuperar humanismo, de recuperar sensibilidad, de recuperar lo esencial del ser humano, que es amar. Dios es amor y nosotros también hemos sido creados por Él para amar. En la Conferencia Episcopal de México, cuando estaba lo más alto en los contagios a causa de COVID, nosotros invitamos a mirar 3 problemas fundamentales y convertirlos también en oportunidades de cambio. La enfermedad, porque puso en vilo todos nuestros sistemas de salud, todos nuestros recursos para poder curarnos, pero también la situación económica de nuestro país, la pobreza extrema, el hambre que muchas personas estaban en riesgo de vivir y desde luego la violencia. Como que esas 3 situaciones se incentivaron, crecieron, se profundizaron. El tema de la enfermedad, el tema de la economía y desde luego de la violencia. Pero como dije, son una oportunidad de transformación, de recuperar humanidad. La Universidad UDEM ha querido siempre poner el ser humano en el centro y creo que la pandemia ha sido también un recordatorio para esta universidad que sirve al hombre y a través del hombre sirve a Dios en desplegar durante el tiempo de pandemia esta conciencia humanitaria frente a la enfermedad, frente al hambre y frente a la violencia. Como dijo el Papa, ojalá que después de esta pandemia no volvamos a lo mismo, sino que regresemos cambiados. Y yo deseo que la UDEM, después de este trago amargo que ha sido la pandemia, regresen con buenos ánimos, con mucho interés, con corazón renovado, con una actitud generosa y solidaria con nuestra sociedad. Creo yo que la pandemia nos ha obligado a abrir nuestras puertas. Es cierto, hicimos confinamiento, nos encerramos, pero en el encierro físico descubrimos también la gran ventana, el portón que se abre para vivir la solidaridad y la caridad. Yo quiero animar, quiero desde luego felicitar a la UDEM por mantener esta cátedra que ha marcado positivamente el caminar de la universidad, junto con la Cátedra de Arte Sacro, para preservar los patrimonios de la humanidad. Y el patrimonio principal de la humanidad es la vida, por lo cual, cuando la vida está en riesgo, en cualquiera de sus circunstancias, la universidad está llamada a desarrollar su capacidad de rescate, su capacidad de protección, su capacidad de promoción. Quiero agradecer esta oportunidad de compartir este sencillo pensamiento, sabiendo que si enfrentamos convenientemente este momento de la pandemia, estaremos creciendo en fidelidad a Dios, en fidelidad al ser humano, a la humanidad y en fidelidad a la naturaleza. El Señor nos pone juntos en esta casa común y en esta respuesta común que tenemos que hacer. Y la Universidad de Monterrey creo yo que ha estado a la altura de la respuesta humana que debe darse en este tiempo de pandemia. Todavía no ha terminado esta crisis, todavía queda un tiempo difícil de recuperarnos, pero creo yo que ya es tiempo también de mirar con esperanza y de recuperar la fuerza interior y el entusiasmo para que la humanidad sea cada vez mejor y más grande. Que la Universidad de Monterrey, siempre generosa, siempre abierta, siempre solidaria, en esta pandemia logre dar un paso cualitativo para pensar siempre en que está al servicio de la sociedad. Esto ha sido muy claro desde que se fundó la universidad, pero creo yo que ahora la pandemia ha recordado tanto a los jóvenes, muchachos y muchachas, como a los directivos, a los maestros y maestras, a todo el personal que colabora con ustedes en la universidad, a los capellanes, a todos, a las congregaciones fundadoras, a todos nos ha puesto en este camino nuevo de esperanza. Creo yo que la respuesta humana hoy tiene un nombre nuevo, que es una respuesta sinodal, es decir, debemos recordar que la pandemia nos ha puesto a caminar juntos, que la Universidad de Monterrey no quede aislada, que se incruste perfectamente en el caminar humano y que esté siempre presente, desde luego, en este lugar, en esta iglesia local de Monterrey, en estos lugares de Nuevo León, pero sabiendo que tiene una vocación nacional y una vocación universal. Que Dios los bendiga. Gracias, Mario Páez, rector de la universidad, por mantener vivo este espíritu y que se conserven estas cátedras, que siempre mi petición, porque estas cátedras visten a la universidad de una característica muy importante, conservadora del patrimonio universal. Primero, el patrimonio de la vida, que es el patrimonio principal, pero también el patrimonio cultural y artístico, que es la Cátedra de Arte Sacro. Que Dios los bendiga y gracias por escucharme.
La Universidad de Monterrey, a través de la Cátedra de Genética y Vida Humana, agradece a Monseñor Rogelio Cabrera, arzobispo de Monterrey, por su valiosa participación. A continuación, cedo la palabra a Eduardo García Luna, vicerrector de Ciencias de la Salud.
Buenas tardes. Buenas tardes.
Buenas tardes, doctor.
Es un gusto saludarlos. Me permito en este momento leer una carta que fue enviada de parte del Vaticano a la señora Florencia Infante de Garza, presidenta y fundadora de la Cátedra de Genética y Vida Humana. Mensaje de su eminencia, el cardenal Gianfranco Ravasi, presidente del Consejo Pontificio de la Cultura, a los participantes de la 20 Cátedra de Genética y Vida Humana, Monterrey, México, 19 de noviembre de 2021. Fechada en Ciudad del Vaticano el 17 de noviembre del 2021. A la estimada señora Florencia Infante, presidenta fundadora de la Cátedra de Genética y Vida Humana. Al saludarles cordialmente, deseo expresarles mi gratitud y admiración por el trabajo que siguen realizando con gran pasión, empeño y dedicación en el ámbito de la Cátedra de Genética y Vida Humana, la cual, en esta su 20 edición, aborda un tema por demás acuciante y trascendental, entendiendo el origen y la evolución de la pandemia SARS-CoV-2. A finales del siglo XIX, el naturista y filósofo Ernest Haeckel popularizó en Alemania el trabajo de Charles Darwin y creó nuevos términos y conceptos, entre ellos uno que sería a la postre preponderante: ecología. Sin embargo, mucho antes de que Haeckel acuñara dicho término, instituciones y organizaciones gubernamentales y no gubernamentales emprendieran la lucha y abanderaran la causa por el cuidado del planeta y del medio ambiente, ya el texto bíblico presenta lo que bien podría llamar los primeros manifiestos ecológicos, de manera que aunque la ecología como tal tiene un origen relativamente moderno, podemos afirmar que hunde ya sus raíces en el Antiguo Testamento. Los relatos bíblicos que leemos en los primeros capítulos del Génesis ponen de manifiesto cómo Dios quiso comunicar su amor llevando a cabo la creación y cómo tuvo el deseo de entrar en comunión con el hombre, formándolo a imagen y semejanza suya, a diferencia del resto de todo lo creado. Asimismo, siguiendo la narrativa del texto de acuerdo con el autor sagrado, encontramos que la existencia humana se basa en tres relaciones fundamentales e interconectadas: la relación con Dios, la relación con el prójimo y la relación con lo creado. Y es en esta última en donde, por el tema que nos ocupa, quiero fijarme. Dios le manda al hombre dominar la tierra, pero este dominio no significa para nada un comportamiento despótico o destructivo sobre la creación, sino que Dios tomó al hombre y lo puso en el jardín del Edén para que cultivara y lo cuidara. Dos verbos que en la expresión original del texto hebreo manifiestan el sentido de la encomienda que Dios da al hombre. El verbo hebreo, abad, que significa cultivar, también significa trabajar, servir, y el verbo hebreo shamar, que traducimos por cuidar, incluye también los significados de proteger, preservar y sostener. En este sentido, ser puesto por Dios en el jardín del Edén para cultivarlo y cuidarlo significa que el hombre fue creado no para explotarlo a placer y sin medida, o por el contrario, para no hacer nada, sino que tiene la misión de trabajar y continuar la obra divina, perfeccionándola y haciéndola prosperar, rindiendo así un verdadero servicio de culto a Dios, cumpliendo su voluntad. Como administrador de grandes bienes, el ser humano no debe pretender ser el dueño. El mundo es una creación de Dios, no del hombre, y el hombre no puede dejar de cumplir esta obligación sin romper su relación de amistad con Dios. Hoy en día, la creación sufre una degradación a gran escala, que no debe dejarnos indiferentes
porque de ello depende la supervivencia de la humanidad, tanto de hoy como de las generaciones futuras. La pandemia ocasionada por el virus SARS-CoV-2 nos ha hecho palpar el drama de la voracidad devastadora del hombre en el uso de los recursos naturales, con sus consiguientes consecuencias. El calentamiento global es perceptible en todos los rincones del planeta. La contaminación del aire y del agua es una tragedia por demás innegable. Las fuertes inundaciones, al mismo tiempo que las grandes sequías y las olas de calor extremo, son ya un escenario recurrente. Y según estudios biológicos, biomédicos y biosociales, acreditados debidamente, las emergencias virales son resultado del dominio antinatural de la especie humana sobre el resto de las formas de vida y la ruptura del equilibrio del planeta. En términos crudos y prácticos, el atentar contra la armonía de la creación es atentar al mismo tiempo envenenando nuestra propia especie y volviendo inhospitable nuestra casa común, olvidando que la salud es un proceso sistémico que incluye el bienestar de la naturaleza y del mundo animal. Pero las tragedias también brindan oportunidades. Junto con los inevitables y dolorosos traumas, la privación de los sentidos y los estímulos externos que nos obligan a las cuales nos obliga la nueva forma de vida, puede favorecer un mejor contacto con nosotros mismos y con nuestro mundo interior, y ampliar así la dimensión de la espiritualidad. Además del seguimiento de una conciencia más atenta a los valores de la vida, puede motivar al compromiso y orientarlo hacia la justicia y lo que realmente importa, tanto en términos de habilidades como de relaciones. De hecho, estoy convencido de que solo a través de un enfoque multidisciplinar y dialógico, orientado al progreso y los avances tecnológicos, pero también y sobre todo a la salvaguarda de la dignidad humana, se podrá afrontar este difícil momento histórico, mirando nuestro futuro con esperanza. El mejor deseo de que la cátedra siga siendo fuente de inspiración y enseñanza para muchos. Quiero felicitarlos por estos 20 años ininterrumpidos de esfuerzo y dedicación en pro de la cultura y la educación. Y deseo reiterarles también la expresión de mi gratitud con la convicción de que con la incansable dirección de la señora Florencia Infante, presidenta y fundadora, y con el importante e insustituible apoyo de las autoridades académicas y administrativas de la ilustre Universidad de Monterrey, continuarán promoviendo el diálogo entre la fe y la ciencia, hoy más que nunca tan necesario. A todos, con mi bendición y estima, firma Gianfranco Cardenal Rabasi, presidente. Hoy, de manera muy especial, en nombre de la UDEM, extiendo nuestro reconocimiento a la señora Florencia Infante de Garza, cuyo entusiasmo y dedicación han hecho posible su instauración. Y compartimos con ustedes un homenaje en honor de su trabajo detrás de la realización de este evento desde sus inicios.
Una trayectoria de entrega plena y fructífera bien podrían definir el paso de la señora Florencia Infante de Garza por la Universidad de Monterrey. es la presidenta y fundadora de la Cátedra de Genética y Vida Humana, que se lleva a cabo en nuestra institución a través de la Vicerrectoría de Ciencias de la Salud y que se caracteriza por ser la única en su tipo, además de contar con aval de la Santa Sede. A 20 años de sus inicios, la cátedra se ha distinguido por contar con la participación de grandes exponentes para tratar temas profundos y un tanto complejos. En este espacio se han conjugado perspectivas tanto científicas como humanas, despertando con ello el interés de varias generaciones dentro y fuera de nuestra comunidad universitaria. La señora Florencia Infante, como principal promotora del proyecto, encontró en la UDEM el espacio ideal para instaurar la cátedra y así hacer rendir sus mayores frutos. Año con año, en la Cátedra de Genética y Vida Humana, han participado prestigiados expositores de todo el mundo, quienes han abordado temas de salud, genética y biotecnología, entre otros, analizados desde un punto de vista ético, científico y católico, a través de expertos en el campo de la ciencia y de la aplicación del conocimiento a la comunidad. Visionaria y certera en sus objetivos, la señora Florencia Infante se distingue por sus valiosas contribuciones a nuestra institución. Por una parte, en la UDEM como presidenta y fundadora de la Cátedra de Genética y Vida Humana y de la Cátedra de Arte Sacro, y por otra, por su sobresaliente labor frente a la Fundación Unidos por el Arte contra el Cáncer, UNAC, así como Fundación Muro.
Gracias por acercarnos a este espacio que nos invita a reflexionar, aprender y enriquecer nuestra perspectiva.
Tu nombre es sinónimo de compromiso y de las grandes cosas que se pueden lograr cuando en un proyecto pones lo mejor de la voluntad, del conocimiento y el corazón.
Gracias por ser un ejemplo de liderazgo y de valor para las mujeres que deseamos hacer un cambio en nuestra comunidad. Conocer tu trabajo nos enorgullece, nos motiva y nos inspira.
Hoy aplaudimos y agradecemos tu valor y tu dedicación en estos primeros 20 años. Deseamos que sigan siempre los éxitos para ti.
¡Muchas felicidades!
Por su destacado compromiso con la comunidad y la educación, así como por su valiosa contribución a la Universidad de Monterrey, reciba nuestro más profundo agradecimiento.
En este momento, invito a Mario Páez, rector de la UDEM, a que nos brinde un mensaje.
Muchas gracias. Estimada señora Florencia Infante de Garza, para la Universidad de Monterrey es un honor contar con su apoyo, experiencia y talento. Celebramos con orgullo su iniciativa y valiosa labor como presidenta y fundadora de la Cátedra de Genética y Vida Humana de nuestra institución durante estas 2 décadas. 20 años que, si bien han presentado desafíos, este evento en todo momento se ha mantenido vigente. La Cátedra de Genética y Vida Humana es un punto en que convergen el conocimiento científico, el humanismo y el pensamiento crítico, los cuales fomentamos en la UDEM a nuestros estudiantes, en quienes buscamos inspirar su mejor versión. Señora Florencia, gracias a su liderazgo, visión y dedicación, durante estos años, hoy tenemos el privilegio de contar con este espacio y grandes expertos que edición tras edición han compartido su experiencia a través de este evento. En nombre de toda nuestra comunidad universitaria, le extiendo nuestra felicitación y nuestro más profundo agradecimiento. A continuación, me permito presentar un mensaje de parte de Álvaro Fernández Garza, presidente del Consejo de la UDEM, a quien hoy no pudo estar con nosotros. Muchas gracias.
En la Universidad de Monterrey nos unimos a la alegría de celebrar un año más de la Cátedra de Genética y Vida Humana. Agradezco la participación de Monseñor Rogelio Cabrera, arzobispo de Monterrey, así como la presencia del doctor Won-Shu Lu y del profesor Dominic Dwyer por formar parte de esta edición. De manera muy especial, en nombre del UDEM, reconocemos el trabajo de Florencia Infante de Garza. Florencia, te felicito por ser la promotora de este gran proyecto, por tu ejemplo y por la huella que has dejado como fundadora y presidenta. Recibe nuestro agradecimiento, nuestro reconocimiento y, por supuesto, nuestra más grande felicitación. La cátedra representa una plataforma de enseñanza que nos ha permitido contar con grandes expertos en temas complejos, que año con año han enriquecido nuestra perspectiva. Desde su primera edición ha dejado en las personas que han formado parte de ella una semilla del conocimiento y de la reflexión, con valor y con responsabilidad. Es importante destacar que los aprendizajes derivados de la cátedra no solo llegan a la academia, sino que trascienden en nuestra comunidad. Para la UDEM, estos 20 años de su impartición representan una enorme gratificación. Gracias por compartirla con nosotros.
Para finalizar este gran evento, cedo la palabra a Eduardo García Luna, vicerrector de Ciencias de la Salud, quien dirigirá un mensaje de clausura y agradecimiento.
Muchas gracias. Como seres humanos estamos llamados a la evolución, al desarrollo de nuestra mejor versión para nosotros mismos y nuestro entorno. La Cátedra de Genética y Vida Humana es una prueba de ello y de cómo, desde un testimonio a una profunda reflexión, puede inspirarnos, llevarnos a la acción y dejar la huella en nosotros como ha sido en esta ocasión. En nombre de la Universidad de Monterrey, extiendo nuestro más sincero agradecimiento a nuestros exponentes invitados de hoy. Monseñor Rogelio Cabrera, arzobispo de Monterrey, el doctor Honshu Lu y al profesor Dominic Doyer, gracias por compartir con nosotros y ayudarnos a comprender a fondo los orígenes y la evolución de la pandemia, tanto desde un punto científico como humano. Es un privilegio contar con la presencia de este tan significativo grupo de expertos para nuestra comunidad universitaria y con la presencia de todos ustedes. Muy amables.
Muchas gracias por sus palabras. A nombre de la Universidad de Monterrey, agradecemos a todos ustedes por acompañarnos. Mi compañera Ana y yo les deseamos que tengan una excelente tarde.